Hello friends, today we are going to discuss about translation inhibitors. Translation inhibitors are inhibitors which stop or slow the process of translation, also known as protein synthesis. They act at the level of ribosome. Ribosome is a site of protein synthesis in both prokaryotes and eukaryotes. These translation inhibitors work at different stages of translation like initiation, elongation, and termination. We can divide these translation inhibitors in three categories. First category is translation inhibitors acting only on prokaryotes. Only on prokaryotes. In this category, five translation inhibitors are there: tetracycline, streptomycin, chloramphenicol, erythromycin, rifamycin. We discuss all of these one by one. So first is tetracycline. Tetracycline obtained from Streptomyces aurofaciens. It binds to 30S ribosomal subunit, यानी कि small subunit. It blocks the binding of amino acyl tRNA to amino acyl tRNA to a site of ribosome. Second is streptomycin. Streptomycin, it obtained from Streptomyces griseus. It binds to 30s subunit. It causes misreading of genetic code, misreading of genetic code, and inhibits inhibits initiation at higher level. So these are tetracycline and streptomycin. Next translation inhibitors, third is chloramphenicol. Chloramphenicol, it obtained from Streptomyces venezuela. It binds to a site of 50s subunit, a site of 50s subunit of ribosome, and it affects proper binding of amino acyl T RNA to the A site, to the A site, and inhibits the peptidyl transferase activity. Now the fourth inhibitor is erythromycin. Erythromycin, it obtained from Streptomyces erythraeus. It binds to 23s rRNA of 50s subunit. It binds in the exit channel and inhibits the elongation of peptide chain. Next, fifth is rifamycin. Rifamycin, it indirectly inhibits protein synthesis. It indirectly inhibits the protein synthesis. It blocks the initiation of RNA chain, in initiation of RNA chain means by binding to RNA polymerase, means it prevent the transcription, and that's why it's inhibit the protein synthesis. So we can write that it prevent transcription, and by inhibit the transcription, it inhibit the protein synthesis. Next category of translation inhibitors is B. Translation inhibitors acting on both prokaryotes and eukaryotes. In this category, we discuss about two translation inhibitors. First is puromycin and second is actinomycin D. So we discuss one by one. First is puromycin. Puromycin, it obtained from Streptomyces albopniger. It prematurely terminates polypeptide synthesis. Second is actinomycin D. Actinomycin D, it binds to DNA. It block the movement of RNA polymerase. It block the movement of RNA polymerase, and that's why it inhibit the synthesis of RNA and protein. This is the second category. Now, third category is C. Translation inhibitors acting on acting on both sorry acting on eukaryotes. In this category, we discuss about four translation inhibitors: cycloheximide, alpha amanitin, diphtheria toxin, and ricin. We discuss one by one. First is cycloheximide. cycloheximide, it block the peptidyl transferase activity. It blocks the peptidyl transferase activity of ATS eukaryotic ribosome. ATS ribosome. Second is alpha amanitin. Alpha amanitin, it block mRNA synthesis by binding to RNA polymerase second. Third is diphtheria toxin. Diphtheria toxin, it catalyzes ADP ribosylation of modified histidine residue of eukaryotic elongation factor EF2, EEF2, eukaryotic elongation factor 2, and inactivate it. Then last, fourth is ricin. Ricin, it toxic protein. It is toxic protein of castor bean. Castor bean is a member of family Euphorbiaceae. It inactivate the 60S subunit by depurination. Depurinating a specific adenosine in 23S rRNA. Okay, so thank you friends.